Hello, thank you for joining us on another episode of the PlantGENE podcast. I am Swadhin Swain, a research fellow at Dartmouth College in New Hampshire. My research is focused on plant hormone signaling and stem cell maintenance. The PlantGENE project centers around the enhancement of plant genetic engineering to ensure a sustainable supply of food, feed, and pipe. In each podcast episode, we welcome esteemed leaders from academia and industry to share their experience and insight for the benefit of our listeners. Please share this space with your fellow plant science enthusiasts. That will help us to bring exciting stories on the frontier research in agtech and plant science to our audience worldwide. Today's guest is Dr. Bill Gordon-Kamm. Bill is a distinguished fellow at Corteva. Bill has worked in industrial biotech for the past 37 years. His research focused on improving transformation of recalcitrant maize inbreds and other cereal crop. So this field is very popularly known as cellular reprogramming. Bill pioneered use of DEB genes or morphogenic genes to increase speed and efficacy of transformation and regeneration of monocot cereal crops. This technology has been widely adopted for many other crop types. Bill is also a member of the PlantGENE Steering Committee. He is helping to create and implement visions for PlantGENE. In the year 2020, Bill received Fellow Award from the Society for In Vitro Biology for his outstanding and significant contributions to the field in vitro biology and demonstrated service to the society. I am delighted that I got this opportunity to talk to Bill. Welcome, Bill. Thank you for joining me today and thank you for your time.
Yes,
Thank you.
I'm happy to be here. Thank you.
Could you please explain what is plant tissue culture and what role it plays in crop improvement?
Sure. So, tissue culture is simply starting with typically small X plants from various plant species and growing them in vitro for various purposes. So, this technique has been widely used for many years for plant propagation and micropropagation of many horticultural crops, many crops that are difficult to reproduce sexually. It is a very integral part of many double haploid protocols, which are widely used in agriculture today for accelerated genetics. And, um, and of course, it provides the platform for transformation and editing protocols for many species.
Is there any example for the like general audience, like one or two examples, like where it is very useful for the society in the last, I don't know, 50 or 30 years? Um, do you have any such example in your mind?
Well, I think, when you look at asexually um, propagated agricultural crops like sugarcane,
Hmm.
it has been extremely valuable in recent uh, gains in the field of editing, where they can't, for example, produce large scale immature embryos for transformation, and so they have to rely on different explants and different techniques of doing their culture for editing purposes, and that's been extremely successful. And I think then probably uh, some of the major agricultural crops such as wheat and maize, uh, double haploid production
Hmm.
relies on in vitro embryo rescue to accelerate the process and identify haploid plants and then take them through the doubling process, typically in vitro, so that they don't have to use these harsh chemicals for chromosome doubling in the field, for example.
So your research focuses on improving transmission and regeneration of cereal crops, and some of these crops are extremely difficult to either transformation and or regeneration. How does your work help to solve the uh, bottlenecks?
So when I started working in maize tissue culture, um, transformation and regeneration were extremely difficult. And improvements in tissue culture techniques traditionally had been done through media manipulation. So through very methodical and painstaking uh, testing, for example, of media grids, hormone grids, to find which combinations of hormones and salts produce the best tissue culture response for a different genotype. In maize, when I started back in the 80s, there was really only one genotype that was amenable to efficient tissue culture and presented a good target for transformation, and that was a hybrid between B73, which is a stiff stock inbred, and A188, which is a agronomically poor inbred, but an inbred that provided some very unique tissue culture properties, very high somatic embryogenesis rates, and it was very easy to manipulate in culture. So that became the first target that everyone really focused on for transformation research in corn. So that type of media tissue culture research was really pioneered by some excellent scientists. Murashige and Skoog were two of the pioneers in that area that really kind of established the techniques for the field and the industry. And I had to do a lot of that type of research myself, but I didn't have the patience for it, honestly. So I've always liked a quote by a science fiction writer, Robert Heinlein, who said that progress isn't made by early risers; it's made by lazy men who want to find easier ways to do things. And I think that probably sums up my research career.
Uh, yeah. Uh, could you please explain like what are the DABE genes or morphogenic genes, and how can someone use them to improve plant transformation and regeneration?
So, as the name implies, these are genes that are very critical at different transitional stages during development of the plant. And two that my lab has focused on. Well, the first one that we focused on was LEC1, which is a gene involved in embryogenesis. And then later we moved on to Wuschel and baby boom, and we tested these individually, and we started testing them in combination. And we found that Wuschel and baby boom together were very effective. At the point that I started using it, baby boom was discovered by a really excellent developmental biologist by the name of Kim Boulier. She discovered baby boom studying microspore embryogenesis in brassica and looking at differential expression between embryogenic and non-embryogenic responses. And baby boom jumped out as being one of the genes involved in very early triggering of embryo formation. Wuschel had been known for quite a while as a gene that was essential for maintenance of meristem structure. And it really is kind of the central gene that signals and controls such things as meristem size. So baby boom wasn't a surprise in terms of being able to use it for embryogenesis. Wuschel, for me, turned out to be more surprising because I expected that ectopic expression of Wuschel would produce de novo shoots. And in many crops, it was found that a pulse of Wuschel expression will actually stimulate embryo formation. Extremely exciting when we first saw it. And so those types of genes, and those aren't the only genes now that are used in this manner. Researchers at University of California, Davis have done some fantastic work with the GRF GIF gene combination.
Yeah, I think
Uh,
it is. Yeah, I think recently Plant Journal Lauren Powell group they published on the maize, right? Just this.
yes, yes, yes. Lawrence Powell's group at Vlam's Institute of Biotechnology and Ghent have done some great work using these types of genes. Um, WOX5 has been published as stimulating improving transformation in wheat and other crops, and recently WOX 2.A. And so has been found to stimulate somatic embryogenesis in maize. So I think there are going to be more genes like this that are going to be useful, and it's just a matter of people being creative and really studying the developmental biology literature and picking and choosing what they think are going to be good candidates and giving them a try.
Right, right. I think you answered my fourth question also about the idea how this Ussel and baby boom came into the scenario. Okay. Would you like to add something more?
Yeah. So as I mentioned, you know, continuing to fiddle around with tissue culture media wasn't my my inclination. It was something that was necessary,
Mhm.
so we paid close attention to it. But I wanted to try to find alternatives, and at in the beginning, when we were trying to work on maize transformation, finding good selectable markers was really difficult. So we were testing out a lot of, herbicides and antibiotics, kind of, the types of resistance genes that you would intuitively turn to. But I became interested in the idea of trying to find plant genes that would stimulate growth. And at the time, cell cycle cancer research
Mhm.
was just booming in the mammalian field and in human research area. And so we started cloning maize cell cycle genes. And we cloned all the maize cell cycle genes we could and tried manipulating them in different ways to stimulate cell division. And in particular, we found that the cyclin Ds and the wheat dwarf virus rep A were very effective in stimulating growth above and beyond normal tissue culture rates. And
Mhm.
so we could use that as a positive selection, but it didn't quite have the power to get us into more recalcitrant genotypes.
Right.
So it worked in things that were already culturable,
Mhm.
but in these really difficult inbreds, such as B73, it wasn't effective. And so that made us start looking in other directions. And morphogenic genes, just immediately when we started seeing these first reports of from Brian Mickey's lab in Ottawa with Kim Boulier and John Harada's lab at UC Davis uh, had some wonderful papers that came out on a number of genes. Um, I mean, it was just too enticing, so we jumped on that area as quickly as we could.
To that point, so when you started just one inbred line of the maize is you can do tissue culture, but how many lines you can do right now at this moment?
So when we started working with Wushel and Baby Boom, we were using a weak promoter for Wushel and then a strong constitutive promoter for Baby Boom.
Mhm.
And with that combination, we found that we could transform about 20% of the inbreds that we tested. And so that was a good start. But then we were always looking for new promoters, and we started concentrating on trying to find promoters that were very highly expressed in the scutellum of the immature embryo.
Mhm.
And we were lucky enough to work with a really fantastic bioinformatician here at Corteva by the name of Mauricio Larota, who found the PLTP gene for us through a lot of filtering and sifting through transcriptome data. in different genotypes. And that PLTP promoter we found gave us initial expression levels that were about 4 to 5-fold stronger than the ubiquitin promoter. And that turned off after 4 or 5 days. So it really just provided a really strong pulse. And then we also switched to an auxin-induced promoter for Wuschel. And so with the auxin levels that we typically use in our culture media, those provided a strong pulse of Wuschel and baby boom. And we found that switching to those promoters then would allow us to, starting with immature embryos, transform any inbred that we tested in maize. Still, the frequencies varied substantially. Some inbreds would give us really high transformation efficiencies, and some were still low, but they were all transformable. And that gave us the flexibility then to start working on genome editing across all of our germplasm. And surprisingly, it also worked in all of the sorghum varieties that we tested, and it worked extremely well for wheat. So it was a very useful combination.
I mean, I'm seeing the literature, people are also using this system for many other crops. Like many other labs have started using this system. Right?
Yes. And then with the PLT promoter, the other very exciting variation on this theme that we found was that if we took 3 viral enhancers and placed them in front of the PLTP promoter, and now use that promoter to drive Wuschel expression, that it produced so much Wuschel in the transgenic cell that cell was no longer regenerable, but it was pumping out so much Wuschel protein, and Wuschel is a diffusible protein, it would move into the neighboring cells and stimulate somatic embryogenesis in its neighbors. And that's an extremely useful method. And we started then using 2 agrobacteria for our transformation, one to deliver Wuschel and one to deliver our trait genes or our editing genes. And we could vary the concentrations of the 2 agros so to skew the outcomes one way or the other, depending on what we were after. And I think that's gonna have a lot of utility in the future for cereal transformation. And then finally, most recently, we've continued to alter the promoters on these genes, and we've developed leaf transformation, which I'm most excited about because it's producing a method that is going to be much more user-friendly and much easier for labs without sophisticated machinery and without the infrastructure to grow donor plants to produce immature embryos. So now you can just grow seedlings in your lab and transform various cereal crops, which I think is going to be extremely useful, and make cereal transformation hopefully much more egalitarian, democratize the method for use across many more academic labs. And we still have a lot of work to do in that area, but it's going well.
I mean, 2 months back, I spoke to Steven Strauss from Oregon State.
Yep.
And he's also mentioned the altruistic method he's using in his lab for the tree plants, for plantation forestry. Yeah.
Yes, his lab is really doing some fantastic research in poplar and eucalyptus. And it's going to be really exciting to see those developments.
Do you have any hypothesis? How do thisel and BBM change the cell fate? Like it is how much it is genetic versus epigenetic? How much, how do they affect the epigenetic landscape, cellular clock? So I'm reading some of the very old literature, like Alan Turing's chemical basis of morphogenesis. Now I understand these are the hormones, which are the morphogenic factor. Then Waddington landscape. So it is now we can take the cell back from differentiated cell to undifferentiated cells. We can go back in the landscape, but still the landscape is very important. So I'm very curious, like how does this thisel and BBM baby boom affect this kind of epigenetic or genetic?
Yeah. So I mean, that's a really exciting question. And I mean, just based on what we know from the literature, um, you know, the genome architecture, um, the epigenetic landscape, when you compare an immature embryo and a leaf cell, have to be radically different from each other.
Okay.
And you know, there's been some really nice work looking at epigenetic changes that occur during development, for example, in maize, and how you know, for example, methylation status around the baby boom locus changes during development. So you know, some of the basic, the framework, the biological framework is known. Um, it's just a matter of someone doing the really careful experiments on how these changes occur during transformation. And um, you know, working in industry, you know, we have to remain pretty focused on practical applications.
Right.
So I just, as much as I've wanted to dig into those areas, we just haven't had the bandwidth to do it. So it really is a, you know, a very exciting area that I hope someone is going to start exploring and really provide some of the answers that you're asking about, because all, you know, all we can do right now is speculate.
All the cells have the same DNA, right? Whether it is a leaf cell or it is a somatic embryo or callus or whatever root, everything is same. But why they are so much different?
Yes.
You know.
Yes.
So
I mean, it's a really fascinating area. Um, and another kind of practical area that I've been involved in for many years is targeted integration. First, through using um, site-specific recombinases such as Cree and the flip frit system.
Mhm.
Um, and then, of course, you know, more recently using meganucleases or Cas9. And, you know, and one of the things that we know from our work is that some genomic locations that we're interested in targeting are more recombinogenic. They're more accessible for integration, either through flip-mediated integration or through homologous recombination.
Right.
And so that has, I mean, most likely that's due to changes in accessibility at that particular location during development, whether you know, you have chromatin condensation going on, or the area is more relaxed and active at a particular time. One of the avenues that I think is still going to be interesting to try to explore is how to relax an integration locus for efficient targeting. You know,
Yeah. Yeah.
that'll be kind of fun.
Yeah. So yeah, I think there are recently at least I saw three publications. One from Danforth's center, they have used recombinase and transposases. They engineer those for the plant. And there are two publications from Arc Institution from they did the same thing in animal cell. I think just last week those publication came out. Yeah, last 2 weeks, I think.
Yeah,
So you don't need to
yeah,
use CRISPR, but you can use the system already exists, but just to engineer those. It's called programmable system. So you can program them and then they can do whatever you want to do on the genome. Yeah,
yeah. Very exciting.
very exciting.
Really cool work.
Yeah, yes. Yeah. So yeah, so coming to the another question, which is maybe interesting for our audience, like what are the impractical scientific idea? Like most of the people believe at some time that these are impossible to do, but only few people believe, okay, we will pursue this and we can do this. So what are those idea in plant science or in ag tech that become practical in your time, like your 40-year career?
Hmm.
And do if you have any example.
Well, so you know, I think we've got a couple examples come to mind for me. The first is efficient targeted integration through homologous recombination. 30 years ago, we've always looked to the mammalian literature to kind of guide us and give us an idea of what to expect in plants.
Mm-hmm.
And in higher eukaryotes, homologous recombination frequencies for people that have been working on trying to do targeted integration have been extremely low. So it's a 1 in 1,000 type of event. So probably the person who's had the most experience in this area over the years is Holger Puchta's lab.
Yes.
Who's just done brilliant work over the years, really pioneered that area.
Right.
But all of his early publications in Arabidopsis, the homologous recombination frequencies were very low. And one pleasant surprise. So our tools have improved dramatically with the advent of the CAS system. So CAS has made targeting much more flexible, but it inherently shouldn't change the recombination frequencies. So, whether a cell is going to predominantly repair through non-homologous end-joining or homologous recombination, is really kind of set by the cellular environment. And, and you can change that. And again, you look at mammalian systems, and it's pretty clear in the mammalian literature that if you have cells that are stuck in the G1 phase of the cell cycle, that non-homologous end-joining is going to predominate. And if cells start cycling more rapidly through S phase and G2, you're going to increase your HR frequencies. And so here we got lucky, because one of the dramatic impacts of Wushel baby boom at the high rates that we're expressing these genes, our cycling time in these small somatic embryos is about 10 to 12 hours. So the cells are just really dividing rapidly, and it provides an excellent environment to do more homologous recombination. And so our frequencies have, they're at levels that we never would have dreamed of. And for me, being able to harness homologous recombination so easily now in plant cells was something that I didn't think was going to be possible. So that was huge. Transforming leaf cells at the efficiency that we can transform them now in corn and other cereal crops was something that I thought would probably never happen. So in my immediate area of work, those are two really impractical things early in my career that have become practical now.
And do you have anything which is impractical right now? Would you like to see them to be practical in the future?
Two of the biggest areas, and there are a lot of people that are working really hard on this right now, are tissue culture-free transformation and editing, which kind of goes hand in hand in a lot of people's mind now with DNA-free editing techniques. So, using RNPs is the simplest example of that. Mm-hmm. But, the development of nanotechnologies that are continuing to improve, and, some of the really clever in planta transformation techniques that people are working on, some of the stuff that's come out of the Voidus lab is providing, some really kind of exciting first glimpses into what we might be able to do in that area. And, it's just going to improve. Tomorrow or the next day, a graduate student's going to wake up with some brilliant new idea.
Yes. I mean, yeah, I mean, tissue culture, and so as the somatic embryogenesis, so there's so much work before you want to do what you want to do, but you have to establish those system. It is so much work, right? You need skilled people,
Yes,
very, very
yes,
skilled
yes.
people. You need to do those stuff. Yeah.
Yeah, it is. I mean, if you take maize transformation and editing as kind of an extreme example, just making the media for doing our experiments for a graduate student to have to come in and make up that media fresh every couple months or, oh my gosh. So, if we could get around that, it would be tremendous.
Yeah. So coming to another topic, I think, so this is something which is in a recent report from the Department of Energy, that the title is like Overcoming Barrier in Plant Transformation, a focus on bioenergy crop. So it was highlighted that lack of talent pool is a major obstacle for plant genetic engineering. So could you please reflect on any action plans that PlantGENE is taking to fill that talent gap?
I you kind of broke up in the middle of that question. I'm
Okay,
sorry. Could
so,
you repeat it for me?
yeah, my question is like the report suggests that that there is a lack of talent pool, that is the major obstacle for plant genetic
Yeah, yeah,
engineering.
yeah.
So my question is, since you are in the steering committee of the PlantGENE, do you have any kind of action plan like PlantGENE? Like I'm asking PlantGENE, does PlantGENE have any kind of action plan to fill that uh, talent pool gap?
This is something that, across industry and academia, it's just become much more difficult to find people that are really interested and excited about this field. So, we're really hoping that um, being able to provide um, this type of virtual interaction to a wide audience is going to hopefully continue to get more people excited. We have a lot of work to do with universities that are working at the undergraduate level to try to um, to get more undergrads excited about plant biotechnology and the opportunities. Um, I one of the big surprises for us. I mean, we had planned on having a series of virtual workshops.
Yeah.
But when we started providing these workshops, it became really clear that it's hard for people to find a lot of the basic information that they need to do this type of work. And so I think a lot of our virtual offering in the future is we're going to try to break this out into modules that we can provide to people, really going back to some of the basics. So we've got one coming up here in the near future on handling agrobacterium. Sounds simple, but there are a lot of tricks and tips that you learn over the years, and we want to make those available to people. So we're hoping those types of things are going to be helpful, and we really want to go beyond just our steering committee and some of the colleagues that we're pulling in. If we can get more volunteers like you who are willing to commit time and effort to helping with this virtual platform, um, maybe it'll continue to spread. I'm hoping that it'll continue to spread,
Yeah.
and we're hoping that it's going to spread, really worldwide, because I think this kind of information exchange, and it's a way for making experts in the community more accessible to students.
Yeah, I was reading that report. Like in US, not that many centers where you can do your plant transformation. And even in our lab, like we want to do rice transformation, it was so expensive. We decided to do by ourselves right now. So we are trying that. So,
Yeah. So yeah, we need to keep working to make these methods more user friendly and more inexpensive. Simplify the methods as much as possible. Um, I mean, I would. So as a first step, I think we can continue to develop the Wishel and baby boom system, focusing on leaf transformation to really make it accessible for a wide range of Poaceae species. And once we've done that, simplify the methods and simplify the media to the point where we could take these media formulations and give them to a company like Phytotech. And so, an undergrad wouldn't have to come in and spend 2 days a month making media. They could just, they could order powdered formulations from Phytotech and make things up a lot more simply. So, I mean, there are so many things that we need to improve. Um, the transformation facilities around the country. I think one of the big problems that they're having is that it's so hard to find and retain the high caliber of employees that they need to run these facilities.
Yep.
It's becoming very prohibitive, and many of the transformation facilities, instead of providing transformation services to anyone who wants them, which was the original model, they're having to fall back and basically just make their transformation services available to the university where they work, which
what happens with the Iowa State? Because the lab used to send to Iowa, but now they are not taking. So we have to send like other places.
yeah, and I hope that's not going to be an increasing trend. So we need to find some way to reverse that. We're going to have to really be creative, I think.
So the last question is, I mean, this is to me, this is a really interesting, exciting time to be a plant scientist. So we would like to like PlantGENE, or our community would like to hear your advice for the next generation plant scientists, what they need to do, I mean, what skill, I don't know, like things they need to do to be successful in their, in the future.
Well, I think be inquisitive, be persistent, really pay attention to the basics. I don't think nowadays you can be, really restrictive in terms of the type of literature that you read. I think you really have to try to develop as broad a spectrum of interest as possible that goes, beyond developmental biology into what's happening in the animal field that's analogous to things that we're interested in doing in plants. What types of new material sciences are available, or what types of new materials are coming out of engineering departments that are going to be useful for uh, in vitro applications. How can methods like 3D printing help us change our tissue culture methods? I think there's so many ways to be creative in this field. You just have to keep your mind really open to possibilities.
Yeah, yeah, yeah. I see the cell programming is a like emerging field. Like you already done those things, but the name was not existed before. But I think now it is called cellular programming, both in animal and plant field. It is so hot. Like you can take a cell and change it to any cell, like any kind of tissues, any kind of thing. And synthetic biology, yeah, yeah, some of the so exciting thing.
Yep.
So, yeah, with that, thank you for your time, and I really appreciate. I think I have more questions, but we can keep that for the next time.
Okay. Well, uh, I hope that this is going to be useful to people, and
This
um,
is
it was fun.
PlantGENE would like to thank the National Science Foundation for their support. PlantGENE is funded through NSF grant IOS 2210962. If you would like to learn more about PlantGENE or become a member, please visit plantgene.sivb.org.